Assalamu alaikum wa rahmatullahi wa barakatuh. Today I'm going to talk about the BRAF gene, its alteration, and how I think we can treat it. BRAF. BRAF is an intracellular kinase that is a protein kinase, which is an enzymatically transferred phosphate group ATP to a serine, threonine, or tyrosine residue. Phosphorylation usually leads in the change of enzyme activity, cellular location, or association with other proteins. This results in the functional changes of target proteins. BRAF's job is to mediate signals from RAS to MEK, ultimately promoting self-proliferation and mobility. BRAF intracellular kinase is frequently mutated in melanoma, thyroid, lung, and other cancers. Fusions. Fusions is one of the alterations of BRAF, which generally form from chromosomal translocations that fuse with the N-terminal end of a partner gene with the C-terminal end of BRAF. The N-terminal is a protein structure with the amino acid that is not part of the peptide bond. The C-terminal is a protein with a carbonyl group that is also not part of the peptide bond. These fusions causes the BRAF kinase to activate mutations. Although not all fusions lead to cancers, sometimes the fusion gene produces fusion proteins which can alter the activity of other genes. In some cases, this alteration leads to those cells growing uncontrollably and form tumors. Chromosome breaks are what lead to fusion. When two chromosomes break, they rearrange parts and fuse together to become two new proteins. This usually ends up in the protein activating cell multiplication. Trametinib and comitinib. Trametinib and comitinib are two drugs used to treat the fusions in melanoma, the most dangerous type of skin cancer. They're both in the class of medications called kinase inhibitors. Since the protein kinase is the one signaling the action of multiplying the cancerous cells, these drugs are perfect since they target these proteins. Trametinib and comitinib both work by blocking an abnormal protein that signals cancerous cells to multiply, which stops the spread of the cancerous cells. Why would I treat fusion to treat cancer? I would rather treat the alteration than just simply taking out the cancer surgically or giving the patient drugs to knock out any other proteins that signal cell mutation, because it's much better to understand the gene and find a drug or make a drug to get rid of the protein itself, and we'd get a cure. How would I treat fusion? We could easily treat this cancer by using the trametinib or comitinib drug and send it into the area of cancer to destroy the proteins activating cancerous cells if the cancer has spread to a great extent. If it hasn't spread a lot, then we can surgically remove the cancer. But this wouldn't stop the formation of proteins that activate the cell multiplication. Fusion oncoproteins are great drug targets since they drive the disease and are only present in cancer cells. But it's very difficult to find drugs that target only fusion oncoproteins. So how could we do that? Primer pair 1 of BRAF has a length of 876. The forward pair and reverse pair both have a length of 20. Exons 9 to 18, which contain the kinase domain, are usually the area where the N-terminal end of a partner gene fuses with the C-terminal end of BRAF. Exons are a segment of DNA or RNA molecule that contains information coding for a protein. We could stop the fusion of the N-terminal end of a partner gene with the C-terminal end of BRAF without hurting the gene. That way, the kinase doesn't activate the mutation of cancerous cells. Perhaps a way to undo the action of fusion, we could possibly send a lot of protein telomeres into the area of fusion. A telomere is the ends of chromosomes which are composed of short repeat sequence and associated proteins that together form a cap. The protein telomere prevents fusion, thus having little of the protein gives the opposite effect, which is why this could work. During cancer, the number of telomeres decreases, so increasing the amount would decrease the number of fusions happening between chromosomes and eventually prevent cancer. Another way we could possibly treat this cancer is using the drug enfuvirtide. Enfuvirtide is a drug that is a fusion inhibitor, meaning it stops the occurring of fusion. Although enfuvirtide is usually used for HIV infections, so the HR1 and HR2 don't interact with each other, which stops them from fusing to host cells, we have a slight chance of the enfuvirtide working even though it's used for HIV infections and not chromosomal fusions.